So, good afternoon, guys. We'll be starting with today's class. So, today we'll be starting with discussion week 8 of the biochemistry course in the NPTL. Okay, so let's start. Okay, so let's start. This is the first question for today. Which of the following reactions release sufficient or more than sufficient free energy required for the conversion of glucose to glucose 6 phosphate? So, what can be the correct answer? Okay, so any guesses? Okay, so B and D. Okay, any more guesses? C. Okay, so let's see the answer. So the correct answer is B, C, and F. Okay, so this is the answer. So first we'll see the different energy changes in different processes and the different steps that takes place in glycolysis basically. Okay, so the first step, glucose to glucose 6 phosphate, that is the step that is in question. So it requires a free energy change of minus 16.7 kilojoule per mole. Okay, so now we have to find out a step that is more than this particular energy value. Okay, so the next step is plus 1.67, but that is not listed in the options. Okay, so it is actually ruled out. Then fructose 6 phosphate to fructose 1,6 biphosphate, this step. Okay, it's actually minus 14.2 kilojoule per mole. That is actually less than the required amount of energy. So this option is actually ruled out. Okay, then moving on to fructose 1,6 biphosphate to DHAP or glyceraldehyde 3 phosphate. This also takes plus 23.9 kilojoule per mole, and this is also not listed in the option. Then conversion of dihydroxyacetone phosphate and glyceraldehyde 3 phosphate, it is plus 7.56. This is plus 7.56. So yes, this is also the correct answer. Then moving on to the different steps. So directly we'll be moving on to phosphoenol pyruvate to pyruvate conversion. That takes around minus 31.4 kilojoule per mole for one molecule of phosphoenol pyruvate to be converted to pyruvate. So that is actually more than the required step. So yes, this is also the answer. Then moving on to the second step, that is ATP. Okay, so if we see, when ATP is actually converted to AMP directly, it releases the amount of energy that is minus 40.6 kilojoule per mole. Okay, so it's actually more than the required amount, that is minus 16.7 kilojoule per mole. So yes, this is also correct. Then we have malate to fumarate. Okay, so it requires an energy change of minus 3.8 kilojoule per mole. So this is actually less than that. So this is also ruled out. Then UDP glucose to glucose 1 phosphate. So in this case, what happens? One molecule of phosphate is transferred from UDP to glucose, forming glucose 1 phosphate. And in this case, the energy change, free energy change, is nearly plus 2.3 kilojoule per mole. So that is actually greater than the required amount. So yes, this is also the answer. So the correct answers would be A, B, C, then F. Okay, these four would be the correct answer. So we'll include A also in this. Okay, so is the explanation clear?
Sir, we will take the negative sign, means we will also take the positive sign and we will also take the negative sign both.
Yes, because like this is like the simple mathematics. When the negative sign is actually we consider it as a more negative, right? So we'll consider that case, and then positive is always more than the negative ones. So that is why we'll consider both.
Sir, but in a negative, the more the value, it will be less.
Hmm. Hmm, hmm.
So in case of phosphoenol pyruvate to pyruvate,
Yes.
it's written minus 30. Oh, okay, okay, twice minus 31
Yes.
point.
Hmm.
Okay, okay, sir.
Point F. Okay. So in, as you can see here, the ATP when that is converted to AMP and pyrophosphate, it releases an energy of 40.6 kilojoule per mole. Okay, so that's why that is greater than the 16.7 kilojoule per mole, that is for glucose conversion to glucose 6 phosphate. So it is also selected.
So, sir, we have to remember these values.
Not really, but you must have an idea. Okay, basically the energy releasing processes, for example, fructose 6 phosphate to fructose 1,6 biphosphate, or phosphoenol pyruvate to pyruvate, these are actually energy releasing processes. Okay, so in that case, the energy change can be greater. So you can have an idea, okay, this energy change is actually greater than the required amount or not. But in other cases, like ATP, you can remember. Okay, because this scenario is actually asked in various questions. But the other ones, you can have an idea. You can not remember the exact value, but you have an idea ki, okay, this can be like in some range, because it's difficult to remember all the values, and this is only the one pathway that we generally study. But you can just have an idea of the major energy releasing steps where substrate level phosphorylation takes place. That will help a lot. And the other, you can have an range or an idea. Okay, so because other steps are not that much energy releasing or consuming steps, so that you can just have an idea, okay, this can be in between. So that can be the case. Okay, so let's move on to the next question. The standard reduction potential for the redox pair NAD plus to NADH, the standard reduction. Oh, wait a minute. Okay, now it's audible. Okay. Yeah, you can just tell me whenever there is an issue because the strength of the signal is really less here. Okay. So the standard reduction potential is minus 0.32 volt in this case, and for oxygen to water conversion, the reduction potential is 0.816 volt. Okay. So for molecule of glucose entering the glycolytic pathway, calculate the free energy change. So you have to calculate delta G with respect to NADH made in the mitochondrial matrix at the end of the ETC. So you have to calculate the free energy change for one glucose molecule. Oh, okay. Wait a minute. Now it's audible. Is it audible now? Okay. So you understood the question, right? Okay. Hmm. I'm not speaking anything. You have to solve the question. So you have to calculate the delta G for for a glucose molecule with respect to NADH, that after it enters the glycolytic pathway completes the Krebs cycle, and then it enters the ETC. So how will you calculate, and what can be the answer? Okay, any answers? So shall we discuss? Okay. Okay, so let's discuss. So, am I audible now?
Yes, sir.
Okay, so first we have to calculate the net reduction potential. Okay, so for that we'll be needing first the reduction potential of the reducing partner and subtracted by the net reduction potential of the oxidizing partner. Okay, so in this case, when ETC happens, oxygen is the major molecule that is actually reduced to water. Okay, so its reduction potential is 0.816 volt. Now, NADH is the major molecule that is actually oxidized in the ETC. Okay, so it will be the oxidizing partner. So the net reduction potential of these two molecules would be 1.136 volt. Okay, this would be a positive value. Now, for delta G calculation, the formula is minus NF reduction potential. Okay, so that is delta E. Just 1 minute. Okay, so now for each of the following reactions, we have two electrons that are involved in the process. So N will be equal to 2, F will be 96500, that is the Faraday constant, and delta E is 1.136. Okay, so after calculating this answer comes out to be nearly minus 220 kilojoule per mole. Okay, now for one, this is only for one NADH molecule that is formed in the Krebs cycle. Okay, so when one glucose molecule enter the glycolysis, it forms two molecule of pyruvate. This is the glycolysis. Okay, two molecules of pyruvate are formed, and through Krebs cycle, one pyruvate molecule actually forms three NADH. So two molecules, if we take for pyruvate, it forms six NADH molecules. Okay, so this delta G will be for six molecules of NADH. So this is for the one molecule of NADH that is being formed in the Krebs cycle. So if we take account of one glucose molecule, that actually forms six NADH molecule through Krebs cycle. Okay, so the final delta G would be minus 1320 kilojoule per mole. So the correct answer would be option B. Okay, is the explanation clear now? Okay, so shall we move to the next question? Okay, so choose the incorrect statement regarding the electron carriers in the ETC. So what can be the correct answer? Any answers? C. Okay, CA. Any more answers? G. So, okay, let's see. The correct answer is A, C, and E. Okay, so let's see. So, first option is lipid soluble ubiquinone acts as an electron carrier in all cell membranes. No. So, it is only present in mitochondria. Okay, so this is a major component in mitochondria that is present as an electron carrier. Okay, in other membranes, ubiquinone might not be act as an electron carrier. Okay, so this is incorrect. Now, porphyrin rings are responsible for the absorption spectrum of cytochromes. Yes, so this is actually a correct answer in this correct statement, because for example this is a iron porphyrin ring that is present in B type cytochromes. Okay, so this ring actually have helps in absorption of light. Okay, so all the this kind of components are involved in the absorption of light in both photosynthesis and oxidative phosphorylation. Okay, then moving on to C, that is cysteine residues are important to form coordinate bonds with Fe ions of heme. So yes, although it's important, but the thing is, mo- uh, more than cysteine, other sulfur molecules are also involved in forming the coordinate bonds. So this is not completely correct, it's partially correct basically. Okay, all the cysteine molecules have their sulfur protruding out, but in this case, not only the cysteine sulfur but also the sulfur molecule also helps in forming a coordinate bond. Okay, so this is partially correct. Okay, but we'll consider it as an incorrect statement. Then 2Fe2S center involves two Fe ions coordinated with four sulfur atoms. Yes, this is correct. So, for example, here So 2FeS. Okay, so here 2Fe is actually being coordinated with 4 sulfur atoms. No, actually it would be 2 sulfur atoms. So this statement is also incorrect. Okay, because in case of 2FeS, for example, here in cytochrome C, the this is rice K iron sulfur protein also. So in this case, 2Fe and 2S are involved. So these yellow ones are the 2S atoms, and this orange ones are the 2Fe atoms. So this means 2Fe molecules are coordinated with 2S molecules. Okay, so both D and E are incorrect. Okay, so here we'll include D also. So A, C, D, E are the incorrect statements of the following statements. So, any doubts? Okay, so D1, because as I said, in rice iron sulfur complex, 2FeS is 2Fe2S is listed. Okay, so that means with 2Fe, two sulfur atoms are actually involved in coordinate bond. But the D statement says that two Fe2S centers involve two Fe ions coordinated with four sulfur atoms. So instead of four sulfur atoms, it would be two sulfur atoms. Okay.
So, but in option, means cytochrome B, there is four sulfur atoms surrounding Fe.
Option B. Okay. So no. One no. The option says that 2Fe2S. What does 2Fe2S means? Okay, so D option states that 2Fe2S is actually being used for two Fe atoms that are bound with four sulfur atoms. But this is not correct, right? Is it fine?
Yes, right.
Okay. So, any more doubts in this question? So, any more doubts in this question? If not, then we'll move on to the next question. So, this is the next question. Cells were cultured and split into three groups, each supplied with cultures rich in biomolecules, vitamins, and nutrients. However, the test groups A, B, and C contain different compositions of plant secondary metabolites. After a 24-hour incubation period, the cells were allowed to recover before being harvested for LCMS analysis to quantify components of ETC and the ratio of cellular metabolites within the matrix compared to the cytosol. The graph below shows the analysis results. Based on the above results, identify the incorrect statements. Okay, so the question says that there were four cultures. Okay, one is the control, and the other three, A, B, and C, are supplied with secondary metabolites that have some role, inhibitory role in the ETC. Okay, and they were grown, and then after that, after the 24 in our incubation period, the cells were now allowed to recover, and they were grown in normal media. Okay, and they were harvested for LCMS analysis, and these four, that is, they check the level for ubiquinol, malate, fumarate, and oxaloacetate in all the four cultures. This is the control one, and this is ABC. So now, based on these results, you have to analyze and then you have to identify the incorrect statement. Any answers? Okay, shall we discuss? So, as the question says, there are three inhibitor secondary metabolites in which the different cultures are grown. Okay. Now, as we see in the control group, the four molecules, ubiquinol is highest, then this is malate, fumarate, and oxaloacetate. Okay. These three molecules are actually involved in the Krebs cycle, and this one is involved in the electron transport chain. So in culture A, ubiquinol is somewhat decreased, but it is actually high. That means ETC is actually functioning fine in case of culture A. But the Krebs cycle and glycolysis might have an effect on the other metabolites. Although malate is fine, but the other two, fumarate and oxaloacetate, had decreased drastically. So that means the Krebs cycle or glycolysis has a major impact in culture A. Okay, now moving on to culture B. In this case, the ETC is actually being disturbed because here the ubiquinol levels are already low. Okay, and the rest of the three are also down, very much decreased. So that means here all the three glycolysis we can actually neglect for now, because here we will not be able to see any of the parts that is actually linked to glycolysis. But yes, indirectly we can say that although if glycolysis is inhibited, Krebs cycle will also get inhibited. Okay, so that's why it is disturbed, and the ETC is also disturbed because the ubiquinol levels are down. Okay, now moving on to culture C, similar thing, but here the fumarate levels are actually high. So, but that is not actually significantly high. Okay, so these in this case also the ETC is disturbed, followed by the Krebs cycle and the glycolysis. So now we'll see the options. Okay, so now option 1 is culture A is in energy surplus state, meaning that it is actually forming energy, but it is not spending its energy. So energy surplus state is actually the body is actually forming more energy as compared to its expenditure. Okay, so culture A is in energy surplus state. This can be seen from the ubiquinol levels. Okay, because ubiquinol levels is high, ETC is actually working fine. So that means ATP production is more, but the other molecules are low. That means this is actually not working. So its expenditure is actually less, because if that will be the case, these molecules, the metabolites of the Krebs cycle, would be high. Okay, so in this case, ATP production is more and ATP expenditure is less. Okay, so culture A is in energy surplus state. So this can be the correct answer. Now, cytosolic concentration of NAD plus will be higher in culture B and C. Okay, so in this case, culture B and C is actually showing low levels of wait a minute. So it is actually showing low levels of Krebs cycle, glycolysis, and ETC. Okay, both the cases. So in that case, NADH formation is actually low. Okay, so in that case, NAD plus concentration will be higher. Okay, so yes, this is also the correct answer. Then culture A contains an inhibitor of complex 2. So culture A is actually here, and it contains an inhibitor of complex 2. No, because complex 2 is actually associated with ubiquinol, right? So if ubiquinol levels are high, that means the complex 2 is also working fine. Okay, so uh, no, sorry. Complex ubiquinol is actually associated with complex 3, that is uh, cytochrome C, cytochrome C and B. Okay, so if complex 2 is actually being suppressed, that means ubiquinone cycle is actually taking place. Okay, so yes, this can be the correct answer. Okay, then glycolysis is inhibited in culture B. So this is actually an extrapolation because the metabolites of the Krebs cycle, that is malate, fumarate, and oxaloacetate, are very less in this case. So we can say that glycolysis is actually inhibited in culture B. That's why Krebs cycle metabolites are also inhibited. Then the flow of electrons is reduced in culture B. So in culture B, because ubiquinol levels are really less as compared to the control group, so yes, flow of electrons, that is ETC, is actually being disturbed here. Okay, so this is also correct. Then proton pumping may have been impeded in culture C. So the proton pumping is actually a case in which the here the proton pumping is actually reduced. Okay, because the electron carriers are already inhibited. So we can say that yes, in culture C the proton pumping may have been impeded, but not really. Okay, because the electrons are actually being moved on, and we do not know at what step this is actually taking place, because ubiquinol levels are low. That means only complex 3 is affected, but the other complexes are not affected. So this cannot be the correct answer. Okay, so the correct answer, that is the incorrect statement, is option F. That means proton pumping may have been impeded in culture C is not the correct explanation. Okay, so any doubts in this question? Okay, if no doubts, then we'll move on to the next question. Cysteine residues are required to coordinate with Fe ions in several complexes of the ETC chain. Which of the following structures will be affected by the UGU cysteine, that codes for 6-cysteine, to UUG, that codes for serine mutation? Okay, so this is a point mutation where the UGU codon is changed to UUG. So what can be the correct answer? There can be more than one option that can be correct in this case. Any answers? Shall we discuss the answer? Okay, so the correct answer is C, D, E, and F. Okay, so yes, heme A and B are actually the porphyrin rings that are attached to the cytochrome molecules. Okay, then we have cytochrome C. Okay, so this is the where is cytochrome C? This is the cytochrome C structure, and here we can see that the S molecules that are appearing are due to the cysteine molecules. So if the this that forms the rice K iron sulfur protein. Okay, so this is the one protein that involves cysteine as an important electron carrier. Then we have ubiquinone. Okay, so ubiquinone is actually involved in the passage of the protons from one side to the other through the ubiquinone cycle, and here also FES proteins play a major role. Okay, then we have NADH dehydrogenase. So here we can see that it has a component of FES, that is basically due to cysteine residues. Then we have succinate dehydrogenase, and it has many FES centers. Okay, so that is involved in electron carrying that have electron carrying capacity. So these four proteins are the proteins that that will be affected when the cysteine molecule is actually being replaced by serine. Okay, so any doubts in this question? Okay, if not, then we'll move on to the next question. So here is another analysis based question. So a new intern mixed the contents of tube containing purified components of the ETC in random order. A senior added NADH, FADH and ATP to these tubes and quantified the products to identify which tube contains which components. Given below is the graph depicting the product levels found after in vitro catalysis. Based on your understanding of the graph and the oxidative phosphorylation, choose the correct statements. Okay, so talking about the graph, there are four tubes, and the person has checked the levels of ubiquinol, oxygen, and the inorganic phosphate. Okay, so based on these analysis, which tube can contain which complex you have to identify. Okay, So please, on the analysis, pick up the correct statements from the following. Okay, shall we discuss? Okay. So C. Okay. Any more answers? Okay. So the correct answer is actually okay. C D E F G I. C D E F G and I. Okay. So the correct answer is A B J and L. So first we'll analyze the graph. Okay. So am I audible? Okay. So first we'll analyze the graph. So in tube 1, so in all the tubes, the experimenter has added all the three: NADH, FADH, and ATP. Okay, and now we have to see, because this is a very vague analysis. Okay, only three components has been analyzed, so there can be a possibility of more than one complex that can be present in each tube. Okay, so in the first tube we have low levels of ubiquinol, oxygen, but high levels of inorganic phosphate. That means ATP production is not here, is not taking place here. Okay, that means this tube can contain complex uh, 1, then 2, and then 4. Okay, yes, and because in complex 5 the ATP synthase is present, so that means inorganic phosphate will be utilized in that case if complex 5 would be present, but that is not the case. So 5 and 3 will not be present here. Okay, then we have tube 2, and ubiquinol is actually the oxidized form of ubiquinol. Okay, so if ubiquinol is actually present, that means complex 3 might not be active. Okay, so these two complexes might not be present in tube 1. Okay, then tube 2, we have low levels of ubiquinol, that means complex 3 is not present here, but high levels of oxygen. Okay, so that means complex 4 will also not present here, because complex 4 is actually helps in reducing oxygen, but here oxygen levels are high, so complex 4 will not be present here. Then same with complex 5, because inorganic phosphate levels are high. So tube 2 can contain complex 1 and complex 2. Okay, now moving on to tube 3, that means ubiquinol levels are high. Okay, so that means complex 3 can be present here. Okay, this oxygen levels are low, that means complex 4 can be present here, and inorganic phosphate levels are low, very low, so that means complex 5 can be present here. And here 1 and 2 might not be present. Okay, then tube 4, ubiquinol levels are low, but that not that much low. So complex 3 might be present. Then there would be a possibility that complex 4 is not present here because the oxygen levels are really high. Okay, so complex 4 is not present here. Then inorganic phosphate levels are also high. That means complex 5 is also not present here. That means complex 3, 1 and 2 might be present in tube 4. Okay, so let's see the options that are here. So tube 1 may not contain cytochrome C, tube 1 may not contain cytochrome B. So these two forms complex 3. Okay, so this 2 might not be present in complex in tube 1. Then tube 1 may not contain complex 2. No, this is incorrect. Okay, because complex 2 can be present here. Ubiquinol levels are low. Okay, then 2 sorry, tube 1 may not contain complex 3. So yes, this is correct. Okay, option D is also correct. Then just 1 minute. Okay, now moving on to the fifth option, tube 2 may not contain complex 1. No, in this case complex 1 can be present, so this is false. Then tube 2 may not contain complex 2. This is also false. Tube 2 may not contain complex 3. Okay, so yes, because ubiquinol is not present here, so complex 3 will not be present here. So this is correct. Then tube 2 may not contain complex 5 also. So yes, this is correct. Okay. Then tube 3 may not contain complex 1 and 2 both. Tube 3 may not contain complex 1 and 2 both. Yes, this is also correct. Then tube 3 may not contain complex 5. No, this is incorrect. Okay. Hmm. So tube 4 may not contain complex 1. Tube 4 may not contain complex 1. No, this is false. Then tube 4 may not contain complex 2. So this is also false. So the correct statements would be A, B, then J and L are false. Then we have D, G, H, and I. So these statements are actually correct. Okay, so is the explanation clear? Okay, so if no doubts, then we'll move on to the next question. Choose the incorrect options from the given statements. So, what can be the correct answer? B. Okay. Any more answers? Okay, so let's discuss. So the correct answer is B, D, and E. So these are not correct. So let's see the options. The electrons from TCA cycle and oxidation of fatty acids enter ETC first at the NADH ubiquinone oxidoreductase step. So yes, the first step in ETC is complex 1, that is NADH oxidoreductase. So from here, the NADH is actually oxidized to NAD plus and the flow of electrons takes place. Okay, so and NADH is actually being produced from Krebs cycle or the TCA cycle and beta oxidation of fatty acids. So these two processes actually lead to production of NADH, and NADH actually enters ETC. So this is the first step in the case of ETC. Okay. Now option B, that is complex 1, consists of lipoproteins and is responsible for reducing NADH. No, it is actually responsible for oxidizing NADH. Okay, so option B is incorrect. Then the path of electrons in complex 2 is succinate to FADH and then to FES. Yes, this is correct. So complex 2 is actually succinate dehydrogenase. Okay, so this has a tendency to convert succinate, and then it directly transfers the electrons to FADH. This FADH transfers the electrons to FES proteins that is present in the structure of succinate dehydrogenase itself. Okay, so the path of the electrons is succinate to FADH and then to FES. So this is correct. Then inhibition of complex 2 reduces the number of protons pumped by the Q cycle from 4 to 2. No, this is false. Okay, because succinate dehydrogenase uh, do not have any link with the Q cycle. Complex 3 is actually linked to the Q cycle. Okay, so that actually increases the number of protons from 2 to 4 in the complex 3. Okay, so this is false. Then 2Fe2S center of the complex 4 is crucial for oxidizing cytochrome C. No, this is false. Okay, because in complex 4, the actually complex 4 is cytochrome AA3. Okay, so this would actually in cytochrome AA3, FES center is involved. Okay, not 2Fe2S center. So that is only involved in the cytochrome CB complex, that is complex 3. Okay, so this is also false. So the correct answer, that means the incorrect statements are B, D, and E. Okay, so any doubts in this question? Okay, so let's move on to the next question. Choose the incorrect statements regarding the alternative oxidase that is present in plants. So what can be the correct answer? Okay. Any more answers? Okay, so the correct answer is option B, C, and D. Okay, so this is the alternative oxidase. Okay, so alternative oxidase is basically this one that actually is present towards the outer leaflet of the inner membrane, which is facing towards the outer membrane of the mitochondria. So what happens here? It can actually transfers the electron from the outside NADH and NADPH molecules that are present, and it can directly transfer it to the cytochrome 4. Okay, and it is actually a cyanide resistant pathway because it can directly act upon with the complex 5, and it can directly supply the electrons to oxygen molecules when cyanide is actually blocking the complex 4, that is cytochrome C oxidase. Okay, so let's see the options now. So option A is NADH is anapleretically generated by light independent reactions in the plants. No, this is false. Okay, alternative QH2 oxidase bypasses complex 3 and 4, mediates the reduction of oxygen. Yes, this is correct. So it can actually directly bypass complex 3 and complex 4, and it can directly transfer the electrons to oxygen. So that when cyanide is actually inhibiting complex 4, then the plant is actually continuing the oxidative phosphorylation. Okay. Then alternative QH2 oxidase is resistant to cyanide. Yes, this is because the cyanide doesn't affect it. Okay, then operation of alternative QH2 oxidase generates heat. Yes, this is fine. Well, this is correct. Then alternative QH2 oxidase accepts electrons from NADH and FADH and directly reduces oxygen. No, it cannot accept the electrons from FADH. It can only accept the electrons from NADH that is actually present in the cytoplasm. Okay, but it cannot use the electrons that are present in the uh, present in the matrix. Okay, so this is false. Then ubiquinone is redundant in alternative QH2 oxidase mediated electron transfer. No, this is also, this can be correct actually, because ubiquinone also acts as a molecule that transfer electrons to the alternative oxidase. Okay, so this is partially correct and partially false. Okay, so the correct answers are B, C, and D. Okay, so any doubts in this question? Okay, if no doubts, then we'll move on to the next question. Choose the correct statements regarding chemiosmosis. What can be the correct answer? A and B. Okay. Any more answers? Okay. So the correct answer is A, C, and E. Okay. So the inner mitochondrial membrane is impermeable to protons. Yes, this is correct. That's why we require the proton pump to pump the protons. Then protons require F1 channels to enter the matrix for ATP synthesis. No, F naught is the pump that is the channel. Okay, that actually helps protons to enter the matrix for ATP synthesis. So this is false. Proton flow during ATP synthesis increases in electronegativity in the intermembrane space. Yes, this is correct. Okay, because the proton gradient is actually being removed there. Okay, so this is the inner membrane. Here the proton accumulation takes place, and through proton pump the H plus are actually transferred to the matrix. Okay, so in that case what happens, the electronegativity here actually increases while resolving the proton gradient. Okay, then alkaline pH of the matrix aids proton flow through the F1 channels. No, this is false. Then inhibition of ATP synthase will halt ETC. Yes, this is correct, because the ATP synthase is actually uh, requires that proton gradient. So that proton gradient will not be formed if the ATP synthesis is inhibited. Okay, because the whole scenario is to generate ATP. So if ATP is already not synthesized, then there is no point of doing the ETC. So that's why it will get halted when ATP synthesis is inhibited. So the correct answer are A, C, and E. Okay. So any doubts in this question? Okay, so let's move on to the next question. Which of the following statements regarding oxidative phosphorylation and ATP synthesis are incorrect? So, what can be the correct answer? Sorry. Okay. Any more answers? Okay, so the correct answer is A, C, and E. So let's see the options. Varenomycin treatment inhibits electron transport by dissipating the proton gradient. So basically, varenomycin is a uncoupler for potassium ions. Okay, so it does not affect the proton gradient as such. So this is incorrect. Addition of oligomycin in vitro will inhibit ATP synthesis. Yes, this is correct. Oligomycin actually affects the F naught complex of the ATP synthase. So it will directly inhibit the F naught molecule. So it will halt the ATP synthesis. So this is correct. Cyanide increases oxygen consumption in vitro. So this is correct because, sorry, this is false because cyanide actually decreases the flow of electrons to oxygen, because it inhibits, the cyanide actually inhibits the cytochrome C oxidase, that is complex 4. So it will not be able to reduce the oxygen molecule, so the oxygen consumption will go down rather than increasing. So this is also false. Then F1 subunit of the ATP synthase can work independently of F0 in vitro. So this is actually correct because the F1 subunit requires only the energy to revolve. Okay, and that energy is actually supplied by F naught molecule. So if we have some in another mechanism to provide energy to F1, so it can function independently of F naught. Then thermogenin mediated ATP hydrolysis produces heat in hibernating mammals for non-shivering thermogenesis. The thermogenin is actually a uncoupler for ATP synthesis. Okay, and it produces heat that is actually called as non-shivering thermogenesis. Okay, but it doesn't have ATP hydrolysis in this. Okay, so this actually takes place in small animals basically, or and it is seen in placental mammals. So this is also false. Okay, so the correct answers are A, C, and E. That are false statements. So is the explanation clear? Okay, so let's move on to the next question. Which of the following statements are false? What can be the correct answer? Okay, so the correct answer is A, C, and D. Okay, so let's see. If a mutation prevents a patient from forming the NADH dehydrogenase complex, the patient can't extract energy from the ETC. No, this is false, because although this is complex 1, but FAD is also formed, FADH is also formed, and secondly, the quinone cycle is also operative. Okay, so these two also help in transporting the H plus and the electrons. So this is a false answer. Although the energy formation will get reduced because the NADH dehydrogenase is not working, but it will not be the case where it will not be able to extract any energy. Okay, so this is false. The role of oxygen in aerobic respiration is the final electron acceptor in the ETC. Yes, this is correct. Then the standard reduction potential of heme iron atom is the same for all cytochromes. No, this is false. Okay, it is different for all the cases. Then iron sulfur proteins can actually participate in one and two electron transfers. No, it is the one electrons that are they help in one electron transport, not two. Okay, so options A, C, and D are the false statements. Okay, so any doubts in this question? Okay, so let's move on to the next question. Which of the following statements are true about the chemiosmotic theory? So, what can be the correct answer? Okay, any more answers? So the correct answers are A, B, and D. Okay, so yes, it was proposed by Peter Mitchell, and this reaction involves both chemical as well as a transport process. Okay, so chemical in the sense it actually involves electrons transfer, and the transport process because it involves the transport of H plus ions. Okay, then blocking on ATP synthesis has no effect on electron transport chain. No, this is false. Okay, because when ATP synthesis is actually blocked, then the proton accumulation takes place that will hamper the equilibrium. So ETC is also blocked. Okay, then only a proton gradient is sufficient to generate ATP. If there is another mechanism of generating that gradient, then ATP synthesis can occur in the absence of electron transfer. Yes, this is correct. Okay, if we are able to generate a proton gradient across the membrane, then that will be able to generate the ATP. Okay, so this is correct. Although if we can use any other mechanism to generate that proton gradient. Okay, so options A, B, and D are correct. Okay, so any doubts in this question? Okay, if not, then we'll move on to the next question. RBCs lose all their organelles during maturation. Which of the following processes can RBCs not use? So, what can be the correct answer? So, RBCs actually don't have any organelles. No organelles are present. So, neither nucleus nor mitochondria nor lysosomes, so that they can make space for the oxygen that they are carrying. Okay, so if there is no mitochondria, that means there will be no oxidative phosphorylation, and there will be no beta oxidation, because both of the things takes place in mitochondria. Okay, so option C and D will not be present in RBCs. All the processes that takes place in the cytoplasm actually operate in RBCs. So be it glycolysis, be it lactic acid fermentation. So these two processes will operate in the RBCs. So the correct answer is option C and D. Okay. So any doubts in this question? Okay, so let's move on to the next question. In the ring in the F naught subunit of ATP synthase can perform 150 revolutions per second. Assuming that it moves constantly at this rate, calculate the time taken for a single ATP synthase complex to produce 1000 ATP molecules. So, what can be the correct answer? Okay, so first thing that you must know is that for one revolution in the F naught, three ATP molecules are formed. Okay, so that means for one ATP molecule, if we see, 1 by 3 revolutions are needed. Okay, secondly, in the question it is given that it performs the F naught 150 revolutions per second. So that means 1 second, in 1 second, 1 by 150 revolutions takes place. And the last thing that we have to calculate is the time taken to produce 1000 ATP molecules. So for one ATP molecule, it is 1 by 3 revolutions. Okay, and in 1 second, 1 by 150 revolutions, and for 1000 ATP. Okay, so this much time would be required to produce 1000 ATP molecules, and that will be equal to 2.22 seconds. So the correct answer is option A. Okay, is the explanation clear? Okay, so let's move on to the next question. A study in rats found that the deficiency of a mineral in the diet causes a decrease in the activities of several proteins involved in the ETC, as this protein contains this mineral. Which of the following minerals could this be? So, what can be the correct answer? Yes, this would be iron, because that is present in Fe, in forming the complex with sulfur of the cysteine molecules. So this is FeS proteins that are present in several ETC membrane proteins. Okay, so let's move on to the next question. What makes 2,4-DNP, that is 2,4-dinitrophenol, work as a weight loss drug? What can be the correct answer? Okay, so the correct answer is option C, and here is the explanation. So this is 2,4-DN, 2,4-dinitrophenol. This one. Okay, so it actually here it has high concentration of H plus ions. Okay, so it binds to the H plus ions and it can directly bring it to the matrix. So it basically stops the ATP synthesis because it is breaking the proton gradient. First thing. Then the second thing is it actually breaks the proton gradient so that ATP synthase will be producing less energy, but the oxygen consumption is actually uh, increased. Okay, so it uncouples oxygen consumption with ATP production so that oxygen is continuously consumed but no ATP is produced. Okay, so that's why it acts as a weight loss drug so that oxygen consumption is increased and the ATP production is similar or less as compared to the normal condition. Okay, is the explanation clear? Okay, so let's move on to the next question. Which of the following is true about the Q cycle, that is the quinone cycle? So what can be the correct answer? Okay, let's see the answer. So it's A and D. Okay, so it basically bridges the switch from two electron transporters to one electron transporter. First thing, because it the thing is it actually transport the electrons from complex 2 and complex 1 to complex 3. Okay, so complex 2 and 1 are two electron transporter and complex 3 are one electron transporter basically. Okay, so it is uh, the bridge between the two electron transporter and one electron transporter. Okay, then the second thing is the net result is the reduction of two cytochrome C. Okay, because from here one H plus is actually transferred and from here one H plus is transferred. So these two are two cycles. Okay, so this one is the first one and this one is the second one. So from both of these, two molecules of cytochrome C are reduced. Okay, so A and D are the correct answers here. Okay, so any doubts in this question? Okay, if not, then we'll move on to the next question. Consider an example where oxidative phosphorylation by a suspension of mitochondrial matrix occurs in a medium of pH 7.4. So, considering this, this is the intermembrane space and this is the matrix. Okay, so the pH of the outer medium, that is the intermembrane space, is 7.4 and that of the matrix is 7.7. Okay, so now we have to first calculate the H plus ion concentration in the matrix. So what can be the correct answer? So that is a numeral value basically. Okay, so the correct answer is 2. That is the hydrogen ion concentration is 2 into 10 to the power minus 8 molar. Okay, so how to calculate it? So basically pH formula is negative logarithm of H plus ion concentration. Okay, and we have to calculate the hydrogen ion concentration in the matrix. So pH of matrix is 7.7. Okay, so now simply what we'll do is we'll put 10 to the power minus 7.7. Okay, so this would give us a value that is 2 into 10 to the power minus 8 molar. Okay, is the explanation clear here? Okay, so let's move on to the next question. This is a similar question. Now you have to calculate the hydrogen ion concentration in the external medium. Okay, so similar to the last question, the pH of the external medium is 7.4. Okay, so this would the hydrogen ion concentration for that medium would be 10 to the power minus 7.4. So this would result in 4 into 10 to the power minus 8 molar. Okay, yes, similar 4 into 10 to the power minus 8 molar. Okay, so let's move on to the next question. Now you have to calculate the free energy difference delta G around this particular membrane. Okay, where you have the temperature to be 25 degrees. So what can be the correct answer? Okay, so the correct answer is 1.7. And how to calculate, we'll see. So this is a membrane. So this is outer membrane, this is inner membrane of mitochondria. Here the pH was 7.7, and here the pH was 7.4. So the hydrogen ion concentration was, for this one was 4 into 10 to the power minus 8, and for this one was 2 into 10 to the power minus 8 molar. Okay, and considering this to be C2 and this to be C1, the hydrogen ion concentration, the delta G is actually calculated as uh, RT ln C2 by C1 plus uh, NF delta psi. Okay, so this is actually the case where we are actually considering the potential energy of the membrane. But in this case, we have neglected this, okay, because the values are not given in the question. We'll only consider this part for the calculation of delta G here. Okay, so delta G, R will be 8.314, temperature is 25 degrees Celsius, so in Kelvin it would be 298 Kelvin. Natural log C2 is actually the more hydrogen ion concentration, that is 4 into 10 to the power minus 8, and C1 is in the matrix, that is less, so 2 into 10 to the power minus 8. This would yield 2. So delta G would be the product of 8.314, 298, and natural logarithm of 2. So this would come around 1717 joule per mole, and converting it to kilojoule, it would be 1.7 kilojoule per mole. Okay, so that's the answer. So any doubts in this question? Okay, if not, then we'll close the session for today. Thank you guys for joining in. Thank you.